Billions of years ago, life on this planet began with single-celled organisms. Now, in the present day, organisms contain millions of cells, each with their own important function. Today, we will focus on the generalized model of the human cell. Welcome, scientists! Today, our journey of self-discovery has brought us to the human cell. What wonders await us inside? What marvels are we to discover as we enter the generalized cell of a human being? Oh my, well it seems we've come to a bit of a blockade. This is the membrane of the human cell. As you can see, not everything can get inside, for it is semi-permeable, consisting of a lipid bilayer with interspersed proteins along it. This layer acts as the cell's first line of defense against dangerous intruders. However, to get inside, we're going to need to prove we're non-hostile. Perhaps carrying something the cell requires could, get us inside. Good thinking! With this bundle of nutrients we can slip inside. Just breathe in that lovely cytoplasm. This fluid acts as the cell's body and can help hold the organelles. Now then, our first stop should be at the mitochondria. We'll need to drop off the nutrients there. You see, the job of the mitochondria is to break down nutrients in order to convert them into adenosine triphosphate, also known as ATP. This is used as the body's energy source. Speaking of producing, look there, it's a ribosome. Looks like a free-roaming ribosome at that. Speaking of ribosomes, there's the rough ER. The rough ER, or rough endoplasmic reticulum, is where we find membrane-bound ribosomes. This organelle's function is to also produce proteins and help with the packaging process. It works closely with the Golgi apparatus to do so. Here the protein will be packaged up for transport outside of the cell. The other half of the endoplasmic reticulum is called the smooth ER. The smooth ER works to produce hormones and lipids. These products can be shipped outside of the cell or used inside of it. Either way, the smooth ER is essential for the production of these bodily necessities. And look, in the distance you can see the nucleus. This is the control center of the cell. Here the nucleus works to maintain the functions of the other cell organelles. It is also where most of the cell's genetic material is stored, serving to protect its replication and preserve it for when the cell splits during mitosis. We'll get back to that in a moment, but first, another organelle associated with protection. This organelle is called a lysosome, and its function is to digest and recycle materials inside of the cell. Any excess materials or unwanted intruders in the cell are absorbed by this organelle and broken down to be recycled and reused. Now that you've learned the functions of the cell organelles, perhaps you'd be interested in learning more about cell replication. I thought so. Well, allow me to explain the process. The first stage of cell division is known as the interphase. In this stage, the cell prepares itself for division by replicating its organelles and copying its DNA. Next is prophase. In this stage, a cell's spindle fibers form on the outer rim of the dividing cells. Metaphase follows after the nuclear membrane breaks down in a process called prometaphase. In metaphase, the now exposed chromosomes line up along the midline of the cell. In anaphase, the chromosomes, also referred to as sister chromatids, are then split apart by the spindle fibers. Then telophase takes place, when the sister chromatids are moved to opposite sides of the cell and the cell membrane begins to separate. Lastly is cytokinesis. Technically not a step in the cell cycle, this is when the two daughter cells form from the splitting of the parent cell, each containing a copy of the cell's chromosomes and organelles. And that is the cell cycle of division. I hope you feel learned in the subject, and I look forward to seeing you in the field of research. Goodbye!